Right, so for our last talk of the fitness session, we have Henrique Figueiró, who's coming from the Vail Institute of Technology, talking to us about conservation genomics in these adorable black-footed ferrets.
Oh, thank you. Here. Yeah. Hi, everyone. It's a pleasure to be here. I would like to thank the organizers for having us here for these amazing talks. And today I'm coming from Brazil, but I'll be talking about pretty much US species, the black-footed ferrets, and some aspects of that we were working for the last couple of years, looking for some genomic aspects about genomic diversity and functional aspects of the species. So a brief outline, I'm going to talk about ferrets, of course, the genetic diversity, some heterozygosity results we have in our analyzing right now, sleep density plots, something that I didn't see through the talks yet, so I think it's a cool result to show you guys. Random homozygosity, which we've been seeing since yesterday, and the demographic analysis that matched with the diversity results we are looking, and finally, like some functional aspects, focus on deleterious variants and the impact in male fertility genes for the species. This work was done mostly through my postdoc within George Mason University, the Smithsonian, with the support of Marine Animal Foundation. And so, what do you know about black-footed ferrets? Before starting, I didn't know much, but it's endemic to North America. Mostly in the United States. There's one recorded population in Canada, one recorded population in Mexico, but most of the distribution is North America. Was considered extinct in the wild in the 70s, and found again like one population was found because a dead individual appeared on a farm. The guy was really clever and told the Fish and Wildlife Service that started a captive breeding program that's happening since then. And so far we have more than 10,000 individuals born in this captive breeding program, which is really successful, but we still like have some problems managing the species, mainly because of reproductive problems. So that's the historical distribution. As you can see, was a huge part of the United States. The introduction sites are in red, and the founding individuals was obtained in Meeteetse, Wyoming, and that's where the captive breeding program started in the 80s. The main threats for the species are habitat loss and fragmentation, declining prairie dog population, and the relationship with synthetic plague, because both species are affected by that. And the black-footed ferrets are really dependent on the environment they occupy and that ecological relation with the prairie dogs. They hunt the prairie dogs, but they also use the burrows to reproduce and live. And the reproduction issues we are seeing right now throughout both in captivity but also for the reintroduced populations are possibly caused by the genetic bottleneck that was caused by the starting a program with just a couple individuals. So our main question is how can we use genomic tools to assess the genetic diversity and specifically the functional aspects to improve conservation programs. Most of the results we are seeing are from wildlife since yesterday, so we are bringing more management of captive and reintroduced species so we can discuss. Our sampling is comprised by 54 individuals, both from the wild, captive and reintroduced populations, ranging from the 70s to the 2010s. We use whole genome sequencing with a coverage of between 10 and 30x. And here in this PCA plot, you can see that the variation is higher on wild individuals compared to the captive ones here, and a little bit more variation in the reintroduced ones. That's a pattern that's going to keep appearing in the other analysis we made. So the genetic diversity, you can see here, the same pattern, wild individuals. Those are like the ferrets from the 70s that were found in museums. Like, they had really good DNA, so we didn't treat them as historical samples, but we used them to have a comparison with the other individuals from the captive and reintroduced populations. And you can see here that their heterozygosity is really small. Even the ones with a little bit better diversity is really small. This plot is not great, but it shows the variation in diversity through time. So from the 70s for our wild populations and our captive and reintroduced ones here. So you can see a drop in diversity through time from the 70s to 2020. And here, like, it's one of our main plots because it shows the mean heterozygosity for our individuals and compare with the runs of homozygosity blocks. As you can see here, it shows the whole genome. So they have like a 2.4, 2.5 gigabases, and approximately 2 gigabases, a little bit less, is made of runs of homozygosity. I'll explain a little more later, but that's the landscape you are looking at. Here's a deep density plot. It's a way of showing the genetic diversity, the amount of SNPs in windows throughout the genome. That's the chromosome 4. And it works like a heat map. So blue is bad, green is okay, red is great for diversity. And you can see it's like different shades of blue for most individuals. Here at the bottom, you can see that the wild individuals from before the captive breeding started, they have lighter shades of blue, but still, like, they have a pretty low density of SNPs throughout the genome. And that pattern goes through every chromosome we look at. Here's another way of displaying these results. That's, instead of looking one chromosome, is the stacked version of that plot that shows the whole genome. And you can see that most of the genome has a low density of variants, and a little bit here, but yeah, and like the red almost doesn't appear here. So we did a zoom here to, we zoom here to take a better look at where the diversity lies in the genome, and we have around 60 megabases of higher diversity in the genome for all our individuals. If you want to see what's going on there, we have only 57 genes in those regions, mostly related to brain function, cell division and organization, immune genes, and some olfactory receptors, which is expected. And they are probably really important genes for maintaining basic functions, so that's why the diversity maybe lies only in those regions.
Genes.
Here's a private allele analysis. So we count the number of singletons for each individual. You can see the diversity and the amount of singletons on the wild individuals are much higher compared to the other ones. We have an outlier here that we are still looking what's going on, but on the reintroduced population, and it's like 60,000 genes more than the rest on average for those individuals. So again, the runs of homozygosity, you can see that we have most of the genome in runs of homozygosity, and I don't like the classification of small, medium, and large, because that changes depending on the species and the things that are happening with the species. But you can see that, like, in Dark orange here are blocks bigger than 225 megabases, and all individuals have like a huge chunk of the genome within blocks of that size. Here is a really famous picture. Everyone read that paper from Ceballos that shows the proportion of the sum of runs homozygosity compared to the number of runs, and this pattern is compatible with a bottleneck and a lot of inbreeding happening, which we can see from our data set. We did some demography analysis in collaboration with Russian PhD student, Ekaterina Doskova. She ran GONE, which is based in LD, so gets the demographic history, the effect number, the effective size for more recent events. So we can see this huge drop in effective population size, really recent, and so you guys can imagine that was when the species was recovered from extinction. So when they found the 7 individuals that started the captive breeding program. And then we want to look the deleterious variants for our data set. We found a lot of high impact genes, more than 1,000 genes related to immune genes and DNA regulation, but also reproduction, which was something kind of expected, but interesting to see. Why fertility is a big problem? Because we see a lot in the captive breeding program, we see a lot of issues with male fertility, sperm motility, mobility, and different metrics of sperm quality. We observe also a decreasing whelping in females. We have amyloidosis problems, neoplasias, some laterality issues with like individuals having just one kidney or just one side of the uterus, which is more rare, but anyway, and that is already described in literature for this species. Male fertility, it's a key fitness trait, and we see an increase in sperm abnormalities for the species, and a decline in pregnancy in females, and it's well documented for ferrets. That's like the number 4 here, that the abnormal sperm increases while the heterozygosity decreases. So that's something we want to have a better idea of what genes are related to that. So using the deleterious variants genes and some fertility databases that were published last year, we are using another one as well. We saw not a lot, but like around 10 genes associated to male fertility that were completely fixed for the 54 ferrets we have sampled, and those are loss of function genes, and they are related to sperm number defects, sperm motility defects, and some other reproductive issues in males. So our next steps is to go further in the past to look if there's when the bottleneck happened, because from our initial data set we can see that the heterozygosity wasn't that good in the past as well. So something was already going on with the ferrets before they almost went extinct. Try to find some correlations between breeding and the phenotype, so looking in more depth for these metrics. Also trying to measure potential load compared to realized load to choose the individuals, and also using some other approach to do the deleterious variants testing. One thing that I didn't mention is that for a captive a species in captivity, maybe the approaches we are using is much more important, and we need to focus on that, because that's where our potential individuals for reintroduction are. So we need to know how is their genomic landscape and evaluate that better. So I'd like to thank our collaborators, the organizers, and the funders for this project. And I'm happy to take questions, and hope you guys are not that hungry.
Amazing. Yeah, thank you. Okay, let's jump into questions.
Oh, it's not on yet.
Okay. Thanks for the talk. This was really interesting. I was wondering about this kind of heat map where you showed like the windows of heterozygosity throughout like the entire throughout the entire genome, and you pointed out, yeah, exactly here, and you pointed out that like the wild one, you know, are slightly more blue, but it looks to be, at least to me, almost like, yeah, it's like mainly the two on top and then maybe like the two towards like the bottom. Can you say something? Is there like, are they from like different places, or is there like a difference between these six or?
Six. Yeah, they come from different populations. Here's Melody County and here's Metitsi, Wyoming. So, and then here is the first couple individuals that started the captive breeding program. So, yeah.
Okay, so the two on top started the captivity program?
Yeah, so that's SB corresponds to study book. So that's study book number 2 and 10. That's one of the first ones.
So they seem to have like more heterozygosity. Was this just like coincidence, lucky, or were they picked like specifically for a certain reason?
Was lucky. It was just like they found the colony with the 7 individuals.
Yeah, and I just want to clarify, SB2 and 10. Sorry.
All right, right.
I can't stop but jump in because I was Henrique's postdoc supervisor. But anyway, SB2 and 10 are cell lines in the San Diego Zoo Wildlife Alliance Frozen Zoo. SB10 created the cloned ferret, the first cloned ferret a couple of years ago, and we recently produced more clones from both of those individuals. So yeah, it's very interesting that SB2 and 10, during their life, they produced no offspring, but their material was turned into a cell line, which was then used through somatic cell nuclear transfer to create that. So, you know, Elizabeth Hand, the first cloned ferret, she was not able to successfully breed, but hopefully this new generation of cloned ferrets will introduce new variation into the population we haven't seen in 40 years. Yeah, and the last two at the bottom, those are from Millette County, like Henrique mentioned, is from Millette County, South Dakota. So that was a completely different population that showed, you know, what we think shows some degree of genetic structure in the species that we don't see in any modern black-footed ferrets. So sorry to jump in,
Yeah,
Henrique.
fine. Thank you.
Thank you very much.
Okay, question over here. Done. Yeah, so I had a question about these peaks of heterozygosity. So it's great that we're here. But it was a great talk, by the way. So in that Channel Island Fox paper, they saw something similar where it was, you know, like the first paper, Jacqueline Robinson's paper, the genomic flatlining. They saw that most of the genome was totally homozygous, and there's these peaks of heterozygosity. And it's actually very coincidental. They also, one of their peaks of heterozygosity was in olfactory genes as well. Mm-hmm. But then they did simulation analyses, and maybe I'm remembering the results wrong, but they did simulation analyses about the distribution and heights of these peaks of heterozygosity, and it could have been totally by neutral processes, as far as I remember. So are you guys planning to look at that to say, like, could this actually be selective, or is this just something that can happen by chance? The amount of these, like, little peaks of heterozygosity.
Yeah. Thanks for the suggestion. That's something we can do. But for olfactory receptors, at least for mammals, it's gonna expect to have that diversity, especially for underground, like a species that spends most of their time underground. There's a lot of diversity on those regions. So I think that that's right. But we are going to take a look on that, and that's your suggestion. Thanks.
have one over here.
Hi, thanks. Great talk. I had a question sort of along similar lines about the more historical drivers of genome-wide genetic diversity. So did you measure longer term any, like through something like PSMC? Because from your GON plots, I would have expected the population to have been much larger, further back, since most of the threats are from plague, which is just like 200 years old since it's been here, and habitat loss and prairie dog declines and stuff.
Yeah, that's another research that is ongoing with Dr. Kepfli group, and I don't remember exactly the pattern, but the DNA was higher in those days, right, Klaus? I'm not lying, right?
We're trying multiple types of these NE analyses because remember we have this problem where the species 80% of the genome
runs a homozygosity, so it's actually really challenging to try to use the remaining diversity to estimate what the effective population sizes, and the 95% confidence interval on that GON curve is very wide. So we're very cautious about this. But yes, we'd like to. We're testing this further. So yeah.
Can we have one?
I think it's flip it up. Yeah, there we go.
Thank you for your talk. I wanted to ask, how have your results here influenced on-the-ground management of black-footed ferrets, especially since you seem to be working so closely with Fish and Wildlife Service?
Yeah, not yet. But that's something we are working on to use those metrics to aid them on deciding on improving the captive breeding program in the future. But we have meetings with them several times to share results and start thinking on the future approaches to use genetics for the pedigree, instead of doing like based on pedigree, using the genomics as a tool.
Any more questions? Okay, then let's
Thanks, guys.
Henrique one more time.